Today's research seminar is from Dr. Matthew Halpert. He is the founder and CEO of Immunocine Cancer Center. He earned his doctorate at the University of Alabama, Birmingham, and then spent 10 years at Baylor College of Medicine in Houston as a cancer immunologist. During his research, Dr. Halpert published research in some of the most prestigious peer-reviewed journals, has been cited over 450 times, and has become one of the leading experts regarding cutting-edge cancer immunotherapy. Additionally, he founded Diagnose Oncology, which is currently studying its revolutionary dendritic cell treatment in multiple FDA clinical trials, including glioblastoma, which was recently awarded a fast-track designation from the FDA in 2023 and won a Breakthrough in Cancer Award in 2024. Other trials are on the same projectile, and there is optimism for future approvals. In 2021, Dr. Halpert left Baylor College of Medicine to found the Immunocine Cancer Center, which leverages the dendritic cell treatment currently being studied in U.S. trials for patients that may benefit today, but are not eligible for one of the trials and do not have the time to wait. He received his PhD in microbiology and immunology from the University of Alabama, Birmingham, and his BS in biology from Southwestern University. Today, he will be sharing a presentation titled Immune Ignorance, Physiological Restoration, and the Benefits in Fighting Cancer. Without further ado, Dr. Matthew Halpert.
Thank you. All right. Well, thank you all for being here on this seminar series, a Zoom call today. I'm excited to share some science with you. I am a scientist. My background is predominantly in immunology and cancer biology. So I am going to get into some science today because I do feel that it is extremely beneficial to understand, or at least have some understanding of the uniqueness here and what dendritic cells can do when used correctly, because they have a really long list of failures and not working. And so this is something that needs to be kind of re-explored. So I'll go through this presentation, kind of go through some of the data, and then we can do Q&A. This is just to be clear, this is predominantly research. I mean, this has been going on for about 20 years. So this initial discovery was about 20 years ago now in the Texas Medical Center with our research group staggering MD Anderson and Baylor College of Medicine. Most of the publications are out of our lab at Baylor College of Medicine. And that's also where one of the trials is ongoing. So let's jump into it if I can. Okay. So just to give some validity, because I am going to obviously get to talking about Immunocine, which effectively operates as a compassionate use setup in Cancun and soon to be in Cabo. And so, yes, that's a little weird because it's in Mexico, but that does not mean there's not validity here or that we're not doing the quote unquote normal pathway things. We do have a variety of FDA clinical trials. We actually have 4 of them at various phases. 2 of them have been fast-tracked by the FDA. Our most advanced is glioblastoma, and we're working on actually publishing that manuscript now. And these are at places like MD Anderson, Baylor, Dana-Farber just came on. So these are, and then we've of course published a variety of times in various peer-reviewed journals about the scientific mechanism. And of course, cancer is difficult, and we're going to talk a little bit about that, but we are purposefully, to move the needle and show what we can do, we are purposefully targeting difficult to treat indications. So again, we've picked glioblastoma, pancreatic ductal adenocarcinoma, angiosarcoma, and refractory melanoma was just picked as our 4th. So we're picking very tricky cancers for the clinical trials. We've seen a variety of early clinical successes. We have I'll show some of that data later. And then we see broad range success. At this point, I think we've treated almost 40 different types of cancer, depending on how you want to break them down. Someday, I am sure we will learn that some cancers respond better than others to this. Today, there would not appear to be a cancer that works or doesn't work, or a mutation that works or doesn't work. And that's largely based on the technology and how we're doing this. So cancer, I'm sure you all have heard, we've all heard over the years, advancements in cancer treatments. Not going to take that away. Obviously, we do continue to move needles. We do continue, there, there new treatments do come out. We do continue to have some sort of success. The trick, though, is often those successes are repeatedly in the same cancers. Some cancers are just simply easier to treat than others. Whereas very difficult cancers, such as glioblastoma, which is actually what's depicted here in A and B, um, or advanced cancers such as late stage, stage 4 cancers, which is the bottom graph. I, you know, that needle hasn't moved for, at this point, decades. In fact, we've been explicitly told by venture capitalists that they don't invest in glioblastoma because that's where treatments go to die. Nothing works. And so for a lot of these really tough cancers, there are not a lot of very good options. And there may not be a ton on the horizon because it's a self-fulfilling prophecy. It's so hard, so no one wants to work on them. The reason we are is largely because we started this as scientists and not business. The business people weren't around when we started, so we went the science route, and um, that does not make them happy, but that is what we did. So why are advanced cancers so difficult? There are really four predominant reasons I want to highlight. They're very diverse. So by the time we get to late stage, they can be very heterogeneous. Occasionally, they effectively are more than one cancer. And so that becomes very problematic for a drug off the shelf to target all of it. And this, of course, can be because those cancers can ultimately mutate away from treatment responsiveness, making the treatment no longer effective. Or, okay, the drug worked. It eliminated the 30% of the cancer that was expressing that receptor and left a void for the rest of the cancer to just fill in. So it buys time, but it doesn't really get the job done. Tumors can be very immunosuppressive. They can hijack immune cells. They can convert them into what effectively are tumor protecting cells. Metastases can be really anywhere throughout the body, and just depending on where they are, they can be very difficult to be reached or um, affected in time. And then, of course, cancer can grow exponentially. And so if it continues to grow and grow and grow rapidly, you have a runaway freight train that has a 95% head start, and the chance of something truly bringing that back is very difficult. I'm going to tell you what we've done to try and, overcome some of this, but I want to be real clear that, to not mislead. We don't have magic. We didn't suddenly cure all cancer. We didn't suddenly, like, it's still cancer. It can still be a very bad situation. Doesn't mean you don't try to fight if you want to fight. I mean, but just to, again, make sure we're being realistic about what we're looking at here, these difficult cancers. So let's move to dendritic cells and why we look at dendritic cells. So dendritic cells were discovered in the 1970s, and they've been known for decades to be effectively the generals of the immune system army. It is their job to see a threat, figure out what the right response is, and create this complete optimal immune response. When you get the flu, dendritic cells, they recognize that, they activate the immune response, and they send the foot soldiers out to eliminate the threat, and not eliminate 50% and then stop, but get rid of it and then have immunological memory and prevent immediate reinfection, or in the case of cancer, relapse. That's what they're supposed to do. And based on biology, a lot of people have tried to use dendritic cells. There was a really big push in the 90s. A lot of money went into, well, let's use dendritic cells to fight cancer, and they really impressively failed. So we'll talk about one of them kind of in particular, but they don't work. And so unfortunately, a couple things happen from that. One, everyone who's gone to medical school or graduate school in the following 30 years have basically been taught dendritic cells cannot work or are not powerful enough to do this. And the market moved and said, okay, if we can't use dendritic cells, what else can we do within the realm of the immune system? And this leads to downstream efforts, predominantly CAR T cell and checkpoint inhibitors, which are artificial and downstream, but powerful and actually do provide evidence that the immune system is capable. So kind of a landmark study was the clinical trial and approval of ipilimumab in 2011, led to ultimately the Nobel Prize for Jim Allison. And so in that example, just removing the brake essentially from immune cells did move the needle for some cancer patients to survive. Not a lot, but some. And the fact that that happened tells us the immune system can do it. The question then is, what is unique about the survivors? How do we get there? What are the missing steps? And so I'm certainly not going to just denigrate CAR T or checkpoint inhibitors. They have their role and they have their place. But again, they are downstream and they're not optimal and they're not complete. So biology does remain constant. That has not changed. And I have not really heard any debate that dendritic cells are kind of at the top of this system. And so what we now know is that 30 years ago, and still to this date, there are some mechanisms within dendritic cells that were being missed. Um, and, to just kind of sound a little bit arrogant, but supported by the data, is this is what we have discovered and resolved. Um, you can have a key that opens a door, but if you just go take that key and slam it randomly into the door, and then the door doesn't open, it seems ridiculous to blame the key. I mean, you didn't use it right. You've got to put it in the right spot or it doesn't go. And so again, for lack of a better term, we found the keyhole. We figured out you have to turn the engine on for the car to go. I mean, it almost is that simple. And when you figure that out, I'm going to show some data to support that. That's what's coming up. But when we figure that out, it changes the narrative. It changes the whole scope. It changes, hold on, we have not viewed dendritic cells or the immune system correctly. So this goes back to, if used correctly, what can be accomplished? So here's a very classical image from any immunology textbook. So I'm taking everyone back to school here. And so if you look on the left, right, you'll see, and if you see my mouse, you'll see like, oh, this is the MHC class 1 pathway where, and just, I'm going to ask this now. Can you all see my mouse, McKenna? Can you all see?
Yeah.
Yes? Okay, I'm seeing
Yes.
a nod. Okay. So, there will be like a viral infection and the virus gets in the cell and proteins are being made, they'll be chopped up, they'll be put on class 1. Class 1 will be presented at the surface and CD8 T cells will respond and you'll get a response. On the other side, you get a class 2 where, bacteria or something is phagocytosed. Ultimately, that's chopped up, put on MHC class 2, and that's presented to a different type of immune cells, the Halpert T cells. And you're going to get different immune responses. And in every textbook that I can see, there's almost always some sort of dashed line or separation, indicating it's one or the other. Either there's going to be this class 1 or this class 2 loading. And dogma says that, dendritic cells will use one or the other. It will launch either a Th1 or a Th2, immune response. And that the dendritic cell is blind, that the dendritic cell doesn't actually know what is on class 1 or 2. It's merely processing, presenting, and the T cell goes to work. Just so we're on the same page, Provenge, which is the only FDA-approved dendritic cell treatment, does neither. It doesn't target either pathway, and it doesn't work. So here's the big discovery that we made, is that this dogmatic schema is wrong. And that dashed line actually should not be there. And in fact, we do know, and others have known, that when there is a viral infection, one of the first things the dendritic cell does is a rapid phagocytic burst, where for 20, 30 minutes, it is just rapidly sampling the extracellular environment. And so the home run here, the big thing here is that, yes, eating bacteria on the outside will lead to a Th2 immune response. But to actually get a complete and optimal Th1 response, you actually need both pathways, not just the class 1. And that has been missed. And it's because this is not the right schema. And what we now know is that there is an opportunity inside the dendritic cell where what is bound to MHC class 1 and MHC class 2 is scanned and compared. And if it is the same, everything changes. And that is not dogma. That is not what anyone thought, and that's not what anyone utilizes. But nonetheless, the dendritic cell puts actually a massive amount of its energy to this program, to this ability to compare and respond. Evolution says it's phenomenally important and conserved and fundamentally responsible for a Th1 immune response. It's not signal 10 in a pathway, it's actually signal 1. And so here's another analogy. People, it's like walking into a dark room and people are trying to flip all these different switches to figure out how to turn the light on. And yes, they flip this switch and on this wall, and maybe they flip two switches and this thing turns a lamp on. And how do we get the lights on in this room? What do we need to do it? And the point is, it's all irrelevant. You're not in the right house. Like you aren't even in the right place in the first place. And that's what has to be fixed. And so when we fix that, so again, not to say maybe there's not another switch that can be flipped, but you've got to start in the right location. So let's go ahead and get to some data. All the data I'm going to show here is published. And so here is just a relatively simple experiment where dendritic cells are loaded, either internal or external, so just one or the other, co-cultured or combined with foot soldiers or T cells. Those T cells then are going to respond. In this assay, the response is measured by the amount of interferon gamma that is secreted. Interferon gamma is a weapon or a tool these cells would use to induce apoptosis and death in, let's say, cancer cells. And everywhere the interferon gamma drops out, red spots show up in the quote-unquote petri dish. And so then the red spots are measured, and that tells you which is working best. And so you can, and so this is what, again, pretty standard. And you look and you say, okay, which dish has more red spots? Is it this one? Is it this one? You know, and because more red spots equals more killing power. And people have played around with, well, let's add this stimulus. And you can picture kind of a margin here of 10% where, yeah, it moves things a little bit. When we enforced this overlap, when we enforced, okay, what happens if we get them to be the same, where we activate this recognition pathway? I mean, it's just, and the y-axis not labeled here is actually fold change, and I need to change that. It's fold. This isn't a 10% shift. This isn't a minor change. It's very significant and very repeatable, to the point where we can't just say 1 plus 1 equals 3. No, 1 plus 1 equals something off the chart. And that's because it's not just an additive thing. It is qualitatively inducing something different. It is flipping a switch. And so when we see this, it makes us say, hold on. If this is possible, then what are we measuring over here? Like, why are we pretending this is more than this? This is all noise. This is just background. If it can do this, we've come nowhere near reaching what a maximal physiological response is. And as a reminder, there's no extra stimulus in this. This is just how the dendritic cells have been loaded. And there's nothing else, no genetic modification. It's just the loading. And so let me take it a step further. And yes, this is what I said. It's not just quantitative, but qualitatively different. And I'm going to also show that in a slide. So let's go to this, because to be clear, it's not just, oh, okay, so you load two things and you get more. Okay, simple enough. It's not just two things. And so let's look at IL-12, which is something dendritic cells secrete. So there's no T cells in this system. So forget the T cells, forget responders. It's the dendritic cell itself. We could load it with like a cancer lysate or a cancer mRNA, and we can measure, yes, here's the IL-12 being secreted. And yes, when we overlap those things, we get this phenomenal and robust difference. But it's not just that there's more, and I'm trying to be very clear about that, because that will, although in a way this is simplistic, that will actually oversimplify it. And so if we add two things, but they're not properly matched up, no, that big response doesn't happen. When we restore the match, it is. And so you can see this purple mRNA is the same across. And no, it has to be the matching correct lysate, not just any. Now, you'll still see a difference in amount. There's always going to be biological variability, People are different, so, but the response is very similar and very repeatable. And again, um, happy to send along publications with a lot more data. Every figure I show here is standing on top of mounds and mounds of data. So I'm going to take this down to kind of a nitty-gritty detail here because this is one of my favorite experiments. So I'm going to try to explain it, but again, not about the what or about the how. So I want you to see these peptide pairs over here. We have a peptide that we know will bind class 1, and you see the sequences here. These are the amino acid sequence, and you see the class 2 peptide, it will bind class 2, and if you look, you see this overlap. WLT, the G's are glycine, etc. We also have this peptide pair, which is pretty much overlapping. WLT, it's the same, except for everywhere there's a G, these two places, there's now an M. So they're almost the same, okay? Just a minor difference in the amino acids, just two of them. And what we're going to do is look at a Western blot here. So again, going back to school. So the way this works is we're looking at a protein being secreted. In this case, this would be something that is pro-Th1 or anti-tumor. The more that is secreted, the thicker the band. So if you see this really faint band from the unloaded dendritic cells, not a lot is being secreted, and they're not loaded. Nothing's there. It's just native background secretion. If we load with just this class 1 glycine peptide, you do get a response. It does increase. Great. And for decades, people have said, yep, there's your Th1 immune response. There you go. That's what we're going to do to kill cancer. And it hasn't worked. It just hasn't. And people still do that today, and it just doesn't work. And so we say, well, hold on, let's load them with both the class 1 and class 2 where they overlap. And we get this is a very significant increase in the amount of this protein being released. And before we say, okay, so again, you gave it 2 peptides, there's more. Okay, well, let's give it 2 peptides, but make sure they don't 100% match. They're gonna be close, but not 100%. And I think you all can already see where this is going, but no, we're back down. We're back down to basically single loaded. And you say, oh, well, maybe the M2, maybe this peptide's messed up. And well, okay, let's restore the match. No, it comes right back. This is so fine-tuned that I can tell you, and what a variety of biologists will tell you, that's not a fluke. This would be the most asinine fluke to exist, where a lot of energy is being used to literally compare the sequences of these bound antigens or peptides or whatever they are, and respond to it and induce a response right away. We haven't gotten to any other stimulation. It's literally just what's there, and are they the same? Are they exactly the same? And by exact, we mean exact. And so it is so, again, and this, for the record, is conserved mouse, dog, human. It's a conserved mechanism. This is how the dendritic cell is meant to work. It is how it is meant to see a threat. And we can, again, talk more about that, but critically important. And though this is a simple experiment and a simple Western blot, it does highlight the, very fine-tuned nature of it, which is why if it's not done right, it doesn't work. And has anyone gone to the trouble in the past 50 years using dendritic cells to ensure this overlap happens? The answer is no. Just no. And so before we get to, I heard a group in Germany uses dendritic cells, or I heard, what about that? They're not, we call this double loading. They're not double loading. We are the only ones double loading. And yes, we have patents on it, we have publications on it, we have FDA trials on it. This is our whole thing. If you do not double load, I don't really care what else you do. You can add BCG or Coley's toxins. I don't know, maybe that does move the needle. It doesn't really matter. You've missed step 1. And so I'm not going to tell you none of those things couldn't be beneficial, but if you do not do step 1, I don't know how you're going to get an optimal response, because obviously the dendritic cell cares. So let me take it further. So that obviously this is my big home run, that I want everyone to leave today with, is how critical it is that dendritic cells are loaded this way. We can also look at the genetic signature of dendritic cells. And so we could take wherever you're sitting, we take a sample of your dendritic cells, And they're going to have genes that we can monitor and measure for immunological, education, activation, power, memory, whatever. We can measure those genes, and they're all going to be baseline. And everyone's baseline is different, whatever. But we'll put that at a value of 0. And if we load that with, mRNA, they're going to change. Absolutely. Some will go up, some will go down. Okay. Same thing with lysate, same thing with other activators. Genes are going to move, they're going to respond. What I'm telling you is that when we double load, the change is not insignificant. And so again, this is published data. And so if you look at this microarray here, unless you're red-green colorblind, I mean, I would assume you can see the difference. A lot of genes change. The dendritic cell chooses on its own to completely change its genetic signature as a result of being loaded this way. And so, again, someone's going to have to look at that and tell me, yeah, but it doesn't matter. Like, okay, if it doesn't matter, then why? I mean, why is the dendritic cell choosing to do that? For the record, almost every gene here, if we get into it, are pro-Th1 immune response. So a lot of that is published as well. Okay, so not about what, but about how. People have tried toxins, they've tried viruses, lysate, peptides, bacteria, mRNA, and a lot of other things. And again, the take home here is it doesn't really matter what it is. It's, the how. And we've published, and we've published this, and we've published this, and we've published this. So it is out there. You can find these papers. You have to correctly double load dendritic cells if you want them to be properly engaged. Because this is such a smart group of people, and this is sort of an educational seminar, I'm actually going to go one step further because as we've talked about, dendritic cells are the generals. They're not the foot soldiers. So how can we explain the actual result we're getting with the killing? Is it just more? Are we just getting more activated T cells and the answer is no. We do get more, but we actually get some unique effector cells as well. And here we go. So we're getting a population of cells which we're calling or have been classified as CD8, CD1, 61 T cells, which scientists have discovered and known about for years. And if you want to go find publications on these cells, you will see that they are enhanced. They are better at homing to solid tissues, such as tumors. They actually are very robust, very powerful inflammatory cells. They have more perforin and granzyme. They're just excellent killers. They absolutely are stronger at killing target, and they have more durability. They actually have much longer life and memory. Scientists have seen these for years and actually written, man, it'd be great if we could figure out where these cells come from and how to use them therapeutically, and no one's been able to. We know they show up during like viral infections and whatnot, but we don't actually know what's stimulating them. And what I'm going to tell you, of course, is that it's the double-loaded dendritic cell that's promoting these. So if you look, this is a dot plot for flow cytometry where we're looking at T cells. And over here in this quadrant are the activated, educated T cells. So antigen-specific T cells, if you're with me. And if we try to stimulate with some random Th1 or use a class 1 or class 2, you see that we're not getting a lot. I mean, you can't even see it. It's less than 1% of the cells are showing up in this quadrant. And again, if we can say, oh, well, we'll do class 1 and class 2, it doesn't make a difference. It's not just that there's more, but when they are properly loaded, that is when we see these cells show up. Again, this is not so much this component is not so much a quantitative difference, but a qualitative difference. We are getting a unique population of cells. And again, this is shown in a variety. It's just one such experiment where conventional T cells are absolutely not keeping the cancer at bay. The cancer in these animals is being lit up. So the more luminescence, the more cancer. CD8 T cells, mix of T cells, or these specific T cells. Obviously, these specific T cells are far more capable at keeping cancer at bay or eliminating cells. And before we jump into some data, just to show immunological memory is so critical if we're going to look and talk about cancer. Obviously, because if you look at palliative care or chemotherapy, and this is a pancreatic cancer, this is a humanized model of pancreatic cancer, also published. Palliative care, pretty much any type of effort will temporarily knock cancer down, and then the patient will lose. That's palliation. Chemotherapy, yeah, sure, it can absolutely knock cancer down, but there's no immunological education, no memory, nothing's been taught. And so if even really a couple cells remain behind, what stops them from just growing out and coming back, stronger than ever? And that is what happened. We see that all the time with chemo, right? What happens if you throw on this vaccine? And the answer is, in this experiment, the cancer did not come back. And not only did it not come back, we couldn't even enforce it to come back. So we actually attempted to just give this cancer to these animals, and we're just unable to do it. The mice were just completely protected in this model. So though, yes, this is a mouse experiment, it is again a humanized mouse experiment, and it elucidated a lot of benefit here. Not to say, for the record, not to say we don't have evidence of success without the chemotherapy, but pointing out that this can be very synergistic or additive to chemotherapy. There's no reason a patient or someone could not do standard of care and this. Quite frankly, everyone should do this. I don't know why you wouldn't want to get your own immune system properly in the game. So I'm going to give you some observations from our glioblastoma clinical trial, which finished enrolling late last year, late last year. In almost all cases, we do see, there's a lot of more data that will be in the publication. This is just representative, but we definitely do see that immune activity tend to increase in these patients. So just looking at various compartments of T cells, the effector cells, they definitely went up post-vaccination. That's not necessarily that surprising at this point, though I'm still surprised sometimes when certain scientists are surprised by that. We actually see local immune activity. So this is a T cell activation marker, and we do see clustering in the glioblastoma tissues after treatment. We were able to get some surgical analysis of that, which was pretty cool. And then I think these are a couple really, really cool MRI images where I'll start with patient 14 here. If you see, and this is a really important point, if you see that there's basically nothing here post-op, and they get the vaccine here at 3 months, and you see this show back up at about 6 months, 8 months. So we'll circle back around, but it's either relapse and progression, or it's pseudo progression. It's not real. It's just activity from immune infiltration. You can't tell based on a scan, and it causes a lot of problems in medicine right now. However, because these patients did not have any clinical symptoms, this doctor said, I'm not going to reoperate, I'm going to let it ride. And over time, you actually see that it went away. And so you see where this presentation's actually 3.5 months old at this point. So fast forward everything 3-4 months, you see that the lesion goes away. This is evidence of an adaptive immune response happening in the brain. Same thing for patient 6. You kind of see this heavy image here at 4 or 5 months, and then it goes away. This is a relapse patient where you see it go away. Admittedly, this is a little late for pseudoprogression. Normally with immunotherapy, it happens sooner. Nonetheless, the data clearly stipulate that that's what it was. It is pseudoprogression, and these patients are doing absolutely fine as of today. So this is the survival curve right now. And I'm going to show 2 points to this. So the group of patients in this, so again, this trial was designed by scientists and not business people. So I'll be honest, we did not do this as wisely as we probably should have, and we anti-cherry picked. Normally you cherry pick and you want the best cases to make your data look better. We went the other way, and 60% of the patients in our trial probably would have been excluded from every other early glioblastoma trial due to very poor prognosis. Importantly, all but one of the patients are unmethylated glioblastoma. So this is the tougher form of glioblastoma, considered not to be treatable really at all. And so median survival is typically about 7.5 months, and almost none of the patients are alive at 14 months. Our first blush here, we have patients now getting out to 2 years and doing apparently just fine. So we definitely moved the needle. So the control line, which should be here, is going to drop down to 50% at 7.5 months. It would be here. Obviously, we've moved that way out here, and that's a great first pass in a clinical trial. But honestly, the data are better than this, and I'm going to explain in this next graph. So about half the patients were with one neurosurgeon, and about half the patients were divided amongst two other neurosurgeons at two institutions. So we had two different institutions and three different doctors. And unfortunately, one of the doctors is not going to come out looking great in this trial, but it's not his fault. He did not know. One neurosurgeon, almost all the patients showed, a lesion reshowing around month 7, 8, 9. No clinical symptoms though, so they're fine. One neurosurgeon said, I don't care if there's no clinical symptoms. Standard of care is to reoperate. I'm going to reoperate. This gray line is for all the patients that got reoperated on, which is about half the patients in the study. Yes, they still got out here the needle is still moved. They still did a lot better, but you can see that at this point, they're all dead. Two other neurosurgeons, including one at the same institution, said, I see the lesion, but the patient is fine. There's no clinical symptoms. I'm going to let it go. That's this line up here, where they're almost all still alive. And yes, this is, again, please move the dots, including this one, 4 months forward. So they're here. In cancer, I mean, it's such a statistically significant difference that it is shocking. You don't normally see such a stark difference in a cancer clinical trial. So I don't necessarily have a phenomenal explanation for this. It's obviously something we did not entirely expect ourselves. It almost looks like going in and reoperating cut out the immune response and changed the battlefield in a way that made the patient susceptible to ultimately losing the fight, whereas the patients that were just left alone have done great. Again, please know this is not even remotely possible in glioblastoma, and so it's almost very surreal data. Anyway, so this will probably be the figure that causes the biggest stir when we publish, because it is effectively saying, please forget standard of care at this juncture, don't reoperate, which is scary, but it's a very big difference. So anyway, this is not a glioblastoma treatment though. Obviously, I've told you it's about the how, not the what. And so there's much broader appeal here than just like, oh, it'll work for GBM. Just to give you some idea. I mean, Immunocine started as a compassionate use offering in Cancun, and it started there on the basis of this is just immunology. It's basic biology. It's very safe. For the record, in the clinical trials, we've had no attributable severe adverse events. It's been very safe, and that's with the brain. We wanted to make this available sooner for patients that can't get into the trial because it, in a way, seems safer than a variety of supplements that a patient could go get. And so why can't a patient get this? Nonetheless, we don't want to jeopardize mainstream movement, working with the FDA, things like that. And so after 3 years of trying to figure out where and how we could do this correctly, we landed in Cancun on the basis of there's an ISO 7 certified positive pressure CGMP compliant lab in Cancun designed for cell therapy. I was very surprised to find that in Cancun, but nonetheless, there it was. And given that it was one of the most certified labs in all of Cancun, the decision was made that this is a lab that could actually do this correctly. So following training of that team, getting the right licenses, getting COFIPRIS on board, getting oncology, radiology, and the right doctors, we launched Immunocine really just as word of mouth. And so for about 18 months, this was word of mouth only. As time has gone on, it's gotten very busy and people have heard about us. And there's been effort to get the word out a little bit more because we think we can really help people. So there's plenty of information, testimonials, things on YouTube, things on our site, Instagram. It's it's getting easier and easier to find us. Just to give you kind of by the numbers, I mean, over 30,000 new website users last year. I would say 2024 is year 2 from a marketing perspective. We started marketing this in 23. So we're still pretty young, but we are getting out there. We do have a limited capacity, so we're working on expanding that, but we're not trying to hide, we're trying to be very upfront and just kind of by the book. At Immunocine, we've treated, this number's again a little, not including the past couple of months because, we are just treating them now. We're at around 100 patients treated with this kind of, our most popular cancers have been breast, colorectal, and prostate, but we've seen a variety of cancers, melanoma, ovarian, bladder. And unfortunately, I mean, I'll tell you now, every melanoma and bladder cancer patient we've treated, which you can see is just a couple, are doing well. I think not every one finally did pass away, and many have reached NED, and we're again talking stage 4. It's just such a small, it's a small number. And so I openly accept that we've got a long ways to go to get to higher N numbers. But again, the data are the data, and that's all I'm presenting here are data. People want to know what are the adverse events. They typically call them side effects. It's an immune response. Most patients, or I guess here half patients, will see fatigue. That's number 1 by far. Tumor-associated pain. It depends where the cancer is. If you have cancer in your prostate, you won't really feel it. If you have it in your spine, yes, you will. Varies patient to patient, but fatigue, pain, flu-like symptoms, maybe fever, chills, headache, things like that. They're usually transient and they're usually grade 1, maybe grade 2. Typically, we're not looking at very severe events. Haven't noticed a particular effect pointing to if a patient is going to pass away or be disease-free. Possibly flu-like symptoms is, a third of our NED patients have had flu-like symptoms, which is a bit more than the other categories. Still a minority of those patients, 2/3 that are NED did not have it, but maybe that will be telling. And so we're continuing to track all this data. If we look at response rates, these are mutually exclusive categories. So if you look at the top graph with no censorship, we have about a 54% beneficial response. Again, this is for stage 4 cancer patients who likely don't have that opportunity. And so that when I've done this data, it just happened to literally be the same number of patients in all 3 categories. I did not know that when I started this, so I promise that's not I didn't make that up. So about 15%, about 18% have seen partial response and are still going. Like, to be clear, that's just where they are today. I don't know where they'll be. About 18% have reached stable and about 18% have reached no evidence of disease, which is pretty high given they had less than a 1% chance. However, for immunotherapy, especially, that's not a really fair way to look at it. And so you will typically see some censorship. So I'm showing you all the data. The censorship is the lower graph, censored for 5 months, and we have a 75% beneficial response at that point. So if you see 50% of the patients that we've treated are reaching stable or no evidence of disease. And when you ask, well, what is censorship? Okay, if a patient comes to us, and so we do decline. I don't think I have in here. We've declined about 85% of applicants. We are not a walk-in clinic. We're not looking to just take people's time and money. So since we've opened, we've declined about 2,200 applicants. I mean, there are easier ways to make money, but that's not what we're doing this for. So if it looks like it might be dangerous, or if it looks like it's not going to be, there's not a good faith belief of efficacy, then we're not going to do it. It's just, again, not ethical to do that. And so we do decline considerably more than we approve. But let's say we approve someone and they pass away. I'll be really ridiculous about it. Let's say they pass away within the week. Okay, technically, that's a patient of ours, and I'm putting that in the top graph. But did that patient really have the opportunity to respond to treatment? Was their immune response really going to ramp up and save them? And the answer is no. And if we could have seen the future, we shouldn't have ever taken this patient because there was never really that chance. And so normally for an immunotherapy, censorship around 4 or 5 months is what you see. Because if a patient doesn't have at least 4 or 5 months, you could debate. We don't know if the treatment worked. They didn't really have time to see what the effects would be. Maybe we've had patients where absolutely, I can tell you, the immune response was educated, activated on fighting. Patient died at month 3. Like there wasn't time. And so if you censor it and say, well, what about the patients that are getting past 5 months? These are our numbers. And so obviously we're getting to, again, I could make up much more impressive numbers. We're curing 90% of patients, but the difference here is these are real statistics. And when I present these usually to oncologists, they're usually pretty impressed. And so censoring out those 5 months, also, a bunch of patients, we've treated them in the past 5 months. So don't count them. And then we have patients that passed away, about 10 passed away within 5 months. And so past 5 months, all time, only 15% of the patients we've treated have actually passed away, which is a pretty low number for the patient population we're seeing. So there's that data. And then here is a graph. So again, how do you put this in a graph when there are so many different types of cancer, different backgrounds, different stage, different chemo? I do have other graphs for specific cancers, colorectal, prostate, but just to kind of try and put it all here, I've got 3 historical curve lines here, just so you can kind of have that in your head. So here's a 50-50 6-month, 12-month, 18-month. So, you can look at whatever line you want. If you want to look at this median survival 12-month, this is kind of the line for if a patient has a cancer where the doctor says, look, you probably have a year. Patient's making the doctor's making the best guess. Obviously, some of those patients don't have a year. They will lose it at 6 months. Others will make it longer. So 50-50. Our lines are here. If you look at the uncensored data, this is where the line is. So this is just all time where we're at with whatever this is, about a 75% survival rate at 30 months. And then if you censor for 4 months, this is where the line are. These dots are not every patient. I just put some of them here so you can see it isn't just one patient that's pushing this curve out, but several are making it pretty far. So again, at some point, obviously, if this story was about one patient with double-loaded dendritic cells, you could say, maybe that's a fluke. But at some point, the data are the data. And a lot of patients seem to be making it further out than they probably were likely given or expected. And again, I don't know what the future holds, but it's hard to say double-loaded dendritic cells are not doing something effective in patients. And based on all the science I showed you before, hopefully that all lines up and makes sense. This is the treatment timeline with us at Immunocine. We can go over more details later, but just to give an idea, the first week is usually kind of the busiest for the patient. There's a lot of labs, baseline scans, and blood work. We got to do a biopsy, so we have to get fresh tumor material from the patient. We have to do apheresis or at least a large peripheral blood draw because we need the patient's immune cells. So that first week is just a lot of baseline reads, meet the team, make sure everything looks good at that juncture, get the immune cells, get the cancer. Then in week 2, we are building the vaccine. We're doing everything in the lab. including all quality testing and things like that. And then the patient will start their treatment at the end of week 2, which is actually an ultrasound-guided injection of those dendritic cells near functional lymph nodes. That is where those go. They should not go in the bloodstream. They don't traffic well from there. They should not go randomly in the belly. They should go near functional lymph nodes. And this is when the immune response is going to start. The patient now has about 2 weeks where it just doesn't really matter if they're in Cancun or back home. I mean, it's in them, it's their immune response. And then we're going to basically repeat at the end of week 4 and the end of week 6, with each one being give or take 3 days, because we're going to do labs, we're going to meet the oncologist, we're going to make sure things still look good. We'll then do the injection, monitor for any emergencies, though dendritic cells historically don't work that way. And this is all hopefully in line to get the patient's immune response in the game, engaged, and on. And so the patient is not needed on site all 6 weeks. They can stay, they absolutely can stay, but they're needed here. And the hope is, of course, we're going to ramp up their immune response, which will start eating away at the cancer. And for some patients, that's literally all they have to do to win the game. Others are going to have to do more. And so continued care is critical. And so to all of you on the call who may be thinking, oh, I know someone or I have a patient, we're never going to say just do this and then go home and eat Cheetos and don't worry about it. That's not what we're saying. You're a cancer patient until you're not a cancer patient. And so keep an eye on things. Let's keep tracking data. We at Immunocine follow our patients. We actually give concierge teams if they need it, because we don't want to lose track of a patient and we don't want the patient to have no guidance at all if they don't have a helpful doctor in their corner. And so checkpoint inhibitors I described before as a boost to an immune response. I do think they're pretty synergistic. We still run the risk of immune exhaustion at late stages. So, hey, after 6 months of fighting, if the immune system starts to get quote unquote tired, how can you boost that? Maybe a checkpoint inhibitor. Maybe you can use low dose metronomic chemotherapy or localized radiation to synergize results. There are obviously evidence-based supplements and alternatives. So long as it's scientifically justified, we're on board with that. We're not going to ever do something or recommend something because someone on YouTube said to do it. But if there's legitimate science behind it, for sure, we're absolutely open to that. Not that it's just, again, a collaborative effort. And so doctors ask us all the time, oh, I'm a big fan of this. What do you think? And hey, it could be very synergistic because the goal is not, look how awesome Immunocine is. The goal is, let's get the patient over cancer. Personally, I think getting their own immune system properly fixed and fighting is a beneficial tool. But absolutely everyone should stay diligent and continue the fight. So wrapping up here, again, dendritic cells are the master regulator of immunity, and people have been trying to use them for a long time. You can stimulate them with different danger signals, whatever, inflammatory stuff or interferons or virus. You can make different dendritic cells. You can make tolerogenic dendritic cells that go the wrong way or do something. You can load them differently. Maybe we use neoantigens. We use these peptides. And again, the point here is all that's fine, but you have to actually load the dendritic cell correctly, and that is unique to us. And I've actually interestingly heard some other group, there's another group that said, oh, we double load. And when asked, oh, what is that? Because double loading is a term we had to make up because this doesn't exist. And we asked them, oh, what do you mean by that? And they didn't know what they meant. They just said, oh, yeah, we do it. And so be careful. That's all I'm going to say, because a lot of these groups will just say what they think they need to say to get a patient down there. I met with a lot of them. Like I know what they're not. I know there's one group that sends out a 2-pager that claims their treatment is FDA approved. It says very clearly FDA approved on the pamphlet. A little deeper dive, what they mean to say is they had a phase 1 IND approved. What that means is they submitted a protocol to do a safety study on their thing. The FDA said, yes, you can do a safety study, which for the record, they did not do, because in their words, it was too hard to work with the FDA. Okay, getting the FDA to say, yes, you can do a phase 1 safety study is not the same thing as saying something is FDA approved. That is, at best, it's phenomenally misleading to the patient who knows no better, and at worst, it's just fraud. And it's very alarming that this is out there because it's and then people will ask us because they trust us, especially if we've declined them. So I feel very bad because we declined them. We said, we don't want to take your money. We don't think this is good. They're looking for something else. We're saying, does this look like a good shot? And it's no, I'm very concerned by the fact that this is how they're marketing their thing because it tells me they don't even necessarily believe in their thing because this is what they're using to get you is a very misleading couple of statements. So I just again caution against red flags and to go with your gut sometimes. Here's some contact information. You may ask, I have a patient in mind, what's the best way to get started? There are two phenomenally easy ways. Maybe two and a half, three. Go to our site, immunocine.com. We have a submission form right there. Anyone can apply. It's not a lot of questions for the patient to apply almost as soon as they do, typically within hours, maybe a day, but typically hours. Our team will reach out to set up the next steps, to set up an intro call and discuss what we're doing. On our site, there is also a physician hub. If you go to that, which has more data and things for physicians, on there is the option to refer a patient. So if you want, you can refer the patient directly. You tell us who it is, give us their contact. Obviously, it's best that you've briefed the patient on this and they're aware of it. But same thing will happen. We will reach out. It has on there, do you want to be involved with this or not? Like, do you want to be contacted as well? But so two ways on the site to simply get into our system and get going. You can also email Nicole Verico is our head patient coordinator. She is the person that will ultimately reach out to the patient and say, hey, yeah, let's talk. Let's see if this is a good fit. And so it's not hard to get started with us. We try to make it very easy. Tons of FAQs on our site. Again, the cost is there, the time is there, the inclusion criteria is there. Like we really try and put everything very front and center, not really trying to be one of those elusive groups. So, but we're trying to help people. And though this, again, it's not magic and we don't cure everyone, you can watch a variety of testimonials on our site to see patients that have, gotten their life back. I mean, it's actually very moving when someone is able to continue on with their life, especially if they're 37 years old or something and have a lot of life left and young kids at home. And so it's just we're fixing biology, we're fixing a biological mistake with biology. And we think that this can come alongside a lot of opportunities, a lot of patients, and really help them. So I am going to stop there. And we'll go ahead and open the floor to, I actually got through that in about an hour. I timed that pretty well. And I'm going to go ahead and take any questions. And I thank everyone for joining and listening and, happy to help as we can.